affected any nucleated cell. It's estimated to affect about a third of the world's population, but despite this, most people who are infected with this parasite don't actually present with symptoms. The main people who are going to present with symptoms are patients who are immunocompromised or individuals who acquired this infection through congenital transmission. Another way that people acquire this specific infection is through eating or drinking contaminated food or water. So there's two main forms of the parasite that we study. The tachyzoids were pictured on the left, and then bradyzoids are pictured on the right. The tachyzoids are the actively replicating form of the parasite, and these are what trigger the immune responses and are responsible for symptoms. Bradyzoids are the latent form of the parasite. They reside within tissue cysts. So we use mice models to study this parasite, and there's two phases of toxoplasma infection. The acute phase is anything that's up to 2 weeks, and at this point, the parasite is within the periphery. Chronic infection is anything that's after 2 weeks, and at this point, the parasite reaches the brain. And that's one way that toxoplasma is different from a lot of different pathogens. It's capable of crossing the blood-brain barrier. Now, we're not exactly sure how the parasite is able to do this, but there's 3 proposed mechanisms. Once the parasite enters the brain, they reside chronically within neurons, and there are 2 peripheral cells that are important in helping control T. gondii levels. They are CD4 and CD8 T cells, and these cells are important because they produce a cytokine called interferon gamma. Interferon gamma is really important in adaptive immune response, and it also produces molecules which kill the parasite cells. We're also interested in studying microglia because microglia are the resident immune cells of the brain. Microglia operate by traveling to sites of infection, and they release immune mediators. Immune mediators just signal for other immune cells to come and travel to the site of infection. Microglia also generate inflammation to protect from foreign invaders. In neurodegenerative diseases, there's prolonged inflammation, and microglia are actually harmful because they end up destroying healthy cells. So through RNA sequencing,
I'm already taping it, Mom. You don't have to.
There it is. It was discovered that microglia take on a disease-associated state during neurodegeneration. So damage-associated microglia, or DAM genes, are associated with an expression of genes, and this expression of genes has been linked to many different neurodegenerative conditions. And in this DAM phenotype, there's an upregulation of microglial markers and then a downregulation of homeostatic microglial genes. And this phenotype has been identified in T. gondii infection. The discovery of this phenotype has raised some questions, like 1, what is the role of DAM genes in infection? And then 2, is this phenotype helpful or harmful? So a particular DAM gene that our lab is interested in is called macrophage expressed gene 1, or MPEG1, because it is highly expressed not only in the whole brain upon infection with T. gondii, but also in macrophages and microglia. So the role of MPEG1 is not fully understood in terms of immunity, but it's hypothesized to be very important in immunity to T. gondii infection. What we do know about MPEG1 is that it's a pore-forming protein, and the formation of this protein is very pH dependent, so it'll only form in slightly acidic conditions. It resides within lysosomes or endosomal compartments. And it's an antimicrobial factor, which just basically means it's been found to kill certain bacterial cells. It plays a role in forming pores in phagolysosomes. And there's a grad student from our lab. She performed an experiment in which she infected wild-type and MPEG knockout mice with this parasite. And then at 6 weeks post-infection, she looked at parasite burden levels. It was hypothesized because we know that MPEG1 is an antimicrobial factor. We hypothesized that there'd be an impaired immune response, and we'd see higher parasite burden levels in the knockout. However, we actually saw the opposite, which is very surprising. So here's some of her data. She looked at Act 1, which is just the gene that measures parasite burden. And here we see in the knockout that the expression is lower. And then she also performed some cyst counts, and again, in the knockout, we see that that is lower as well. And this was a really interesting and rare finding because it's really rare for the cyst to go down like this. So it raised a lot of questions. Like, we wanted to see if this immune response was better in the brain. And these results also led to my project, which was aiming to answer the question, do we observe differences in T. gondii ability to reach and infect the brain during during acute infection? So how we went about this, we infected wild type and MPEG knockout mice with the parasite, and then we performed qPCR on brain and peritoneum samples, and then we performed ELISAs using blood serum, and we collected these samples at 8 and 12 days post-infection, 8 days because that's when the parasite begins to infiltrate the brain, and then at 12 days because that's when it begins to infiltrate immune cells. And I did forget to mention earlier, qPCR, that's just going to be used to measure the parasite burden, and then ELISA assays to measure the levels of interferon gamma. So at 8 days post-infection in both the brain and the peritoneum, we didn't see a difference in parasite levels, which means the ability of the immune system to control the parasite is the same in both cases. And if you look, you see the peritoneum, the levels are a lot higher here, and that's because the parasite has yet to reach the brain. It's still traveling to the brain. All right. And then looking at our interferon serum, interferon gamma, the ELISA assay, it's really good to measure interferon gamma levels in blood serum because that's a good way of making sure that the immune system is assessing how the immune system is controlling our parasite. And as I mentioned earlier, interferon stasignoline is really important in making sure there's not a fatal amount of parasite in the brain. And as you can see here, the levels are the same. We don't see a difference in the wild type or the knockout. And then 12 days post-infection of the brain, we once again don't see a difference between the wild type and the knockout, but it does appear slightly lower, so there's a potential trend for a decrease in the MPEG knockout, which suggests that something might be controlling the parasite burden in the brain, or starting to. So overall, we didn't see any differences and T. gondii's ability to infect the brain during acute infection. So based on those results, we think the parasite level that we saw at 6 weeks post-infection, that control, that difference that we see, it might be caused by better parasite control that happens later on in infection. So in the future, we would like to observe parasite burden at a later time point, so maybe 3 or 4 weeks post-infection. And then also we're interested in looking at how this parasite levels are affected in other damaging piece. So I would like to thank the Harris Lab for welcoming me for the summer and being willing to answer so many questions. You guys have helped mold me a lot into the researcher I am right now. And two of my students in the lab are actually here right now, Sydney and Isaac. Guys, I'd like to thank Nadia, my mentor, for being here too. And then Janice and Adrian for making all of this possible, and for everyone who listened in person and online.
Still good.
She blew it. I mean, just
Any questions?
any questions?
Okay, right. So it's mostly gonna present as neuro or ocular problems, and it can kind of affect behavior. Some people who have been infected with toxoplasma have
She gave thanks to all
actually been found to have
nice people.
schizophrenia.
If you got
And then there's
questions,
also
answer them.
something called toxoplasmic encephalitis, which is where tachyzoites and bradyzoites, those forms I showed you like way back on the second slide, can actually kind of convert. So, bradyzoites can convert to tachyzoites due to like changes in pH or shock proteins or temperature. And when that happens, there's a proliferation because there's all those parasites within that tissue cyst that breaks down. Then you have an excessive amount of parasites in the brain, which can cause inflammation. And it's something that happens in immunocompromised patients, and it can actually be fatal.
Question. I mean, like the parasites like this and pathogens, I mean, if I go back to do something to try to fight them off, a lot of them is sort of hijack, some of the body systems to actually facilitate both their activity and pathogens. Do you think this could be one of those genes, or like these other pathogens might be quite important?
Oh wait, did you get that last part again?
Oh, do you think that like the gene you were looking at might be one of those genes that the pathogens actually using take advantage of to sort of facilitate this pathogenicity, and do you think So the other guarantees are also,
I'm not really sure, but I think that's definitely something we're looking into in the future.
all right, any other questions?
Are there any treatments or is the infection like for life?
The treatment is pretty much for life if you get it. I'm pretty sure, if I'm remembering correctly, for people who are immunocompromised, there's certain medications